I'll start with our forward-looking statements. Of course, we have forward-looking statements to disclose. First, I'd like to start off with an overview of Caribou Biosciences. Underpinning our technology is a novel next-generation gene editing technology called Chardon-a. It's far more specific than first-generation CRISPR and allows multiplex editing with high genomic integrity. Today, we're using Chardon-a for our pipeline of wholly owned, off-the-shelf CAR-T therapies, and we're using edits to armor cells to potentially improve anti-tumor activity. We're doing that with checkpoint disruption, immune cloaking, and cytokine support, and we're advancing 4 programs in hematological malignancies and solid tumors. We feel we're well-resourced with over $400 million on our balance sheet. That should lead us to Q4 2025. So here I'd like to highlight Caribou's pipeline of CAR-T and CAR-NK therapies. We have 3 CAR-T therapies for hematological malignancies. CB10 and CB11 are in phase 1 studies currently. And we also have an allo CAR-NK platform edited from iPSC cells. I appreciate the opportunity today to highlight the underlying technology and key updates from this year. We'll spend particular time on CB10, our lead candidate, especially with data released earlier this year that demonstrated CB10 can drive durable responses that rival autologous CAR-Ts. I'd like to dive in first and introduce you to Chardon-a, Caribou's novel genome editing technology, and then explain how we use Chardon-a to make our allo CAR-Ts and CAR-NKs. So what is Chardon-a? Besides being a reference to Caribou's Northern California location near the wine country, it stands for CRISPR hybrid RNA DNA, the novel guides that were invented at Caribou. It turns out that inclusion of DNA into the guide dramatically improves specificity to targeted sequences and resulting editing. It enables quite sophisticated multiplex editing while maintaining genomic integrity. It's also highly versatile and made available via standard chemical synthesis. But don't only take my word on it, and let's look at some data. On the left, we looked at on and off target editing for Cas9 with either Chardon-a or an all RNA guide. Both guides were designed to hit the exact same target, a gene for PD-1. But both dark blue and light blue showed high on-target editing. But unfortunately, the standard all RNA guide is also quite adept at off-target editing, which we don't want in gene editing, of course. So Chardon-a shows no off-target editing, at least to the limits of detection of the assay used. In the middle, we see the same effect with a Chardon-a with Cas12a, where we have high on-target editing and non-detectable off-target editing. Finally, on the right, we know that knocking out a gene is one part of the puzzle. We need to knock in the genes that we desire as well. And here, targeted insertion of genes is critical. We actually are looking at a batch of our second candidate, CB11 on the right, at GMP, showing efficiency of gene insertion. It's close to 80% for insertions and upwards of higher than 63% for both edits. This is really incredible editing. I want to shift the focus from the Chardon-a gene editing technology to the anti-tumor effect these edits focus on. We can be quite sophisticated at armoring our cells. And fundamentally, at Caribou, we believe the future of cell therapy has to be off the shelf to unlock these therapies to a broader patient population. We deploy multiple distinctive strategies to armor our cells. The lead candidate in blue, CB10, benefits from checkpoint disruption by the PD-1 gene knockout. The goal is to take the brakes off the CAR-T cells to prevent T-cell exhaustion and keep in a high anti-tumor state as long as possible before immune-mediated rejection. CB11 in green deploys a strategy to immune cloak the cells in multiple myeloma to prevent rapid rejection by the patient's immune system. Of course, the PD-1 and immune cloaking strategies could be combined, and that's exactly what we've done with CB12 in teal. I should also point out that Caribou retains all rights to all these programs in all therapeutic settings. Now, I would like to shift the focus to CB10, our lead program, which was designed as a single-dose, off-the-shelf CD19 for relapsed refractory B-cell non-Hodgkin's lymphoma. It's currently being evaluated in an ongoing phase 1 trial. Earlier this year, dose escalation I'll show shortly, we released dose escalation data that I'll show shortly. It's manufactured using Chardon-a and Cas9 and has 3 edits: knockout of the TRAC locus to remove the T-cell receptor and reduce the risk of graft versus host disease, site-specific insertion of the CD19 CAR, and a knockout of the PD-1 gene to prevent premature T-cell exhaustion. In preclinical data that we have on our website, it suggests that the PD-1 knockout potentially improves therapeutic index relative to other CARs with this knockout. And we believe we've seen this effect in the clinic as well. CB10 is being evaluated in an ongoing phase 1 trial that we've dubbed ANTLER. There are two portions, the dose escalation, which has been completed, and the current ongoing dose expansion. The dose escalation portion had 16 patients at the 40 million, 80 million, and 120 million CAR T cell doses. Today we're currently actively enrolling second-line LBCL patients in an ongoing data expansion. I'd like to provide, before getting to the safety and efficacy data, I'd like to provide a quick look at the 16 patients that enrolled in the study. 10 had LBCL and 6 had other forms of aggressive B-cell non-Hodgkin's lymphoma. Now first, looking at safety, we see that CB10 has a generally well-tolerated safety profile, and I'd like to dig into the 3 adverse events of significant interest on the right. That's CRS, ICANS, and infections. We saw 2 of 16 grade 3 ICANS, 1 grade 3 infection, and no CRS. When compared to the initial pivotal trials that led to the approval of Kymriah, Yescarta, and Breyanzi below that, we can see the data from the ANTLER dose escalation study compared quite favorably. Of course, we acknowledge this was based on 16 patients and not the 100 to 200 in each of the studies that led to the approval of those therapies. But again, our approach is off the shelf, so there's a difference there. Now we move to the efficacy data, which was released over the summer. The swimmer plot here shows the efficacy for all 16 patients, and very excitedly, we saw a 94% overall response rate out of these 16 patients. 69% achieved a complete response, and perhaps the most important number of this presentation is the durability. We saw 44% of patients have a CR at 6 months or beyond. So we were very excited to see that number. In fact, the first patient that we enrolled at the very top has a CR out to 24 months already from 1 dose of CB10. As I did before in safety, I'd like to compare CB10 in terms of efficacy to the approved autologous CAR Ts. When looking at ORR, CR, and most importantly, CR at 6 months or beyond, CB10 compares quite favorably to auto CAR Ts. These data demonstrate that CB10 can lead to durable responses relative to approved autologous therapies. To summarize this section, CB10 efficacy rivals autologous CAR T. It generally has a well-tolerated safety profile, and as an allogeneic therapy, it's available off the shelf. It has RMAT and Fast Track designation, and next steps are to encourage the FDA to evaluate a pivotal study. We expect feedback sometime this year. Shifting now to CB11, Caribou's second allogeneic CAR T therapy and ongoing phase 1 study. CB11 is the program that sparked Pfizer's interest in a $25 million investment in Caribou announced earlier this summer. For that, Pfizer has a 30-day right of first negotiation for this program. Caribou, again, retains all rights to this program in hematology and other settings. CB11 is manufactured with Chardon-a and Cas12a. The edits here are a knockout of the TRAC locus to get rid of the T-cell receptor, site-specifically insert a BCMA-specific CAR, and deploy an immune cloaking strategy that revolves around 2 edits. The first is a knockout of the beta-2 microglobulin, or B2M gene for short, and then site-specifically insert a transgene that fuses B2M to HLA-E. Here we see a representation of the effects of these edits of CB11 and their purpose. The B2M knockout gets rid of the endogenous HLA class 1 presentation in one fell swoop. That's really the goal of the edit. It stops T cells from recognizing and rejecting therapy. However, to complete the armoring strategy, we need to address the missing self hypothesis that cells would be susceptible to without. In particular, NK cells, which are part of the innate immune system, are specifically tuned to recognize and reject cell armoring class 1 HLA. So we take the extra step to insert B2M HLA-E transgene. The goal is for the cells to have HLA-E, but no other major biomarkers on the surface, to prevent rejection by T cells and NK cells. I want to be clear, the goal was not to construct cells that could entirely avoid the immune response and system, but to buy additional time for anti-tumor activity. CB11 is now being evaluated in an ongoing phase 1 study that we've called Camouflage for relapse refractory multiple myeloma. The design is similar to ANTLER, a 3+3 approach. Dose level 1 at 50 million cells is done, and we're currently enrolling dose level 2 at 150 million CAR T cells. No specific timelines for a clinical update have been announced, as the data is event-driven. I'd like to wrap up now with CB12, Caribou's third program, this one for relapse refractory AML. It's also manufactured using Cas12a and Chardon-a and benefits from a PD-1 knockout, similar to CB10, the B2M knockout immune cloaking approach that we saw in CB11, and to our knowledge, this is the first time that both strategies have been deployed together in one clinical program. CB12 is targeted to CLL1, which is an important antigen in AML. It's highly expressed on AML tumor cells and leukemic stem cells, but not healthy hematopoietic stem cells. It allows us to develop a potentially disease-modifying therapy in AML. Just like other programs, CB12 is off the shelf, one time, and wholly owned by Caribou. This program is on track for an IND before the end of this year. Finally, I'd like to conclude with a few additional points. Progress on CB10 is ongoing with dose escalation data that rivals autologous CAR T. The dose expansion is ongoing, with data expected in the first half of 2024. We are currently planning to engage the FDA in discussions for a potential pivotal second-line study, and we'll share feedback potentially before the end of the year. In CB11, dose level 1 is done, and patients have been safely dosed. On CB12, we released data earlier this year at AACR. I encourage you all to peruse our website to find that or contact me. And we plan an IND submission before the end of the year. And again, we feel we are well capitalized with over $400 million on our balance sheet and a runway that we believe is extended until the end of Q4 of 2025. Thank you very much.